Thanks very much for the opportunity to present here. And today we'll be talking about community conservatism. Now, evolution leads to certain ecological patterns in all kinds of organisms. Related organisms tend to be found in similar environments. This is called niche conservatism, and they generally resemble one another. So they share similar traits, and they resemble each other morphologically. An example for this is illustrated here, where you can see the leaf shape of oak trees. If you compare to closely related species, their leaf shape is very similar, whereas if you compare it to something that's further away on the phylogenetic tree, it is less similar. Now, these patterns are well established in animals and plants, and they help us to understand crucial aspects of speciation, evolution, and how the species might adapt to rising challenges such as climate change. Now, we do not only have animals and plants on our planet, but we live in a microbial world. Microbes are everywhere, they drive biogeochemical cycles, and as we heard before from great talks from Chris and Lindsay, they're important for bee microbiomes and human health and disease. But despite their importance, critical aspects of bacterial evolution and ecology remain unknown. So how does it look? What do we know about niche conservatism and phylogenetic signal in microbes? Generally, it is assumed to be there as well, but it is way more difficult to prove and quantify this. Despite this, it is used in many popular algorithms, such as UNIFAC, for instance. Why is it so difficult to prove? Well, many bacteria are still uncultured, their genome is not sequenced, so we don't really know exactly what they look like or which traits do they possess. So we wanted to tackle this question from a little bit of a different angle. And instead of focusing on what we do not know or what we cannot say for certain about microbes, we looked at what information do we have at scale. And what we do know is with the rise of new sequencing strategies and more sampling be done in all kinds of environments on our planet, are the communities in which the microbes occur. And we here hypothesize that we can take this community composition as a proxy for the niche and or functional potential of a microbe. So to summarize this in one question, we want to answer whether or not two close related species, OTUs, are found in similar communities, similar microbial communities to one another. And in order to answer this, we need a lot of data. And this is where the Microbe Atlas project comes into play. This is a database and resource that was recently published around a month ago. And it is a project by our whole lab where we downloaded and re-annotated, reanalyzed all publicly available sequencing samples. We mapped them to a full-length reference database of 6S and 18S RNA sequences, and then obtained a global OTU table that is annotated with different geographic and environmental variables, metadata, and is also accessible as an interactive web resource, which you're welcome to check out after the talks of today. And this now gives us the possibility to trace microbes around the globe and see exactly where they're found, with whom they're found. And we take this as a basis, and I'm going to illustrate my workflow with 4 example species. So first we check with the Microbe Atlas project in which samples are these microbes found. And then in this case, we're looking at 4 different species or OTUs. We're looking at 2 Streptococcus sanguinis subclades. They are very closely related, belong to the same species, and are found in the oral microbiome. So we would expect their communities to be very, very similar to one another. On the other hand, we have Campylobacillus clostridium, which is a generalist species, also sometimes found in the mouth, but generally, or was first detected in yeast, and belongs to the same family, so also the Bacillota. And then as a last species, we have SAR-324, a very cryptic bacterium that's found in the deep sea ocean hydrothermal vents. So we would expect them to basically share no community members with the other species because they're found in a very, very distinct environment. And then what we do is that we compare always pairs of these OTUs in a pairwise manner. In this case, Streptococcus and Campylobacillus. And we do always compare 2 main parameters. One, we compare the similarity of the communities in which they're found. So we're looking in which communities is OTU1 found, and then we look at which samples or communities is OTU2 found, and we compare all the samples of OTU1 against all the samples with OTU2, calculating the Berg-Curtis similarities. And in the end, we average this out, and then get one value that tells us how similar are their communities to one another. And on the second, as a second parameter, we look at the relatedness. So how closely related is OTU1 with OTU2? And for this, we simply take the phylogenetic distance, or the tree branch length, from a high-quality phylogenetic tree of all the bacteria that we have. And in the end, we end up plotting these pairs on a scatter plot, where on the y-axis we have the similarity of the communities, and on the x-axis we have the phylogenetic distance. So in this small example, we see that this trend is present, which we then call community conservatism. So a pair of Streptococcus on the very left side, so very closely related, is also very, very similar in their communities. With a member of the same family, it is less closely related, and also their communities are more dissimilar, but still share some members. Whereas if we compare Streptococcus to SARS-3-4, they are very distantly related only, and also their communities are very dissimilar. Now, this, of course, is only a very small example. But how does it look like if we extend this with all the microbial diversity that we have? And we now repeated this with 25,000 different pairs. And each single dot here is, again, one of these OTU pairs with the two parameters, like I just explained. And they are furthermore annotated based on the latest taxonomy that the OTU pairs share. So if they belong to the same species, they're colored in purple, to the same genus in green, and so on. The red line on the bottom is the null model. So how much do the samples, or how much on average are the communities similar to one another? And what we can see is that with increasing relatedness, also their community similarity becomes more and more similar. This already starts picking up here at the phylum level and then rises almost exponentially. So to answer our previous question with one small sentence, yes, we can show that community conservatism, or the trend of related microbes to be found in more similar communities, is present in microorganisms. And if we bin this according to the latest taxonomy and analyze this statistically, we can actually show that there's traceable signals back until the phylum level, which split up from one another billions of years ago. So this means that despite their frequent, their global presence, horizontal gene transfer, and all other methods of spreading around, we can actually trace back the signal, and bacteria and archaea pass on their community preferences already for billions of years. Now, in the interest of time, I will just show you very, very quickly on one slide some of the other things we did to show that this signal is now actually present in all investigated phylum environments. So first, we saw that there's a very big environmental effect. So a lot of the OTUs that are closely related also share the same main environment. So they're both, for example, found in ocean or soil, but it is also present if we compare it with different environments. Then we saw that all phyla that we analyzed, which were around 20 archaeal and bacterial phyla, showed a signal with different strengths and shape of the trend line. And then we went ahead and we showed that this shape of the trend line actually is corresponding to the habitat, generalists, and specialists. And then going back to the phyla curves again, we can show which phyla are presumably more generalist or more specialist. The details of this are then also in the publication. And I want to show you one more point, which is the outliers. So far, I've mostly presented to you the trends that behave as expected, right? This all hopefully made sense. But what if we look at the pairs that are not behaving as expected? And I actually believe that these are some of the most interesting results that we have. Because for instance, if we go to the bottom left corner, we have pairs that are very closely related, in this case, two Pseudomonas clades, but they're still very different in their environmental communities. And this can show us which species are in the process of really speciating or evolving into different niches. So for instance, here we have Pseudomonas, that is a very generalist, but another clade that appears to be found pretty much only in cystic fibrosis, so in the human lung. And on the other hand, if we have pairs that are very distantly related to one another, such as Streptococcus and Haemophilus, they are still found in very similar communities, in this case, also in the human oral human microbiome. And in this case, these two species have actually been shown to interact and to even form biofilms together. But while this pair is known, we have a lot of them that are not known, and that we provide as a list to find new potential pairs of interacting microbes. And as a very last point, maybe as a teaser, what we think this could be useful for is that I think it this has to do with how we view a species in microbes. So usually what we do, and which is very pragmatic and works reasonably well, is we just look at some kind of marker gene or genome similarity, and we put things into a bin based on their similarity of this marker gene. But this completely ignores all ecological parameters, and of course is not an ideal situation. So in this hypothetical example, we would take purely ecological similarity, where we would cluster OTUs based on how similar their environments or the communities are to one another. And we think that in the future, we could actually, and an example for such a signal would be this community conservatism, or the similarity of their communities. And we think that by combining these two approaches, so taking a purely sequence similarity-based metric such as 16S similarity, but then also considering the average pairwise community similarity of these different species, that we could result in a more ecologically informed OTU clustering, which would then, for example, group things together that are very similar in terms of the communities, but split apart closely related species that are actually doing very different things. All right. And with this, I'm at the end. I hope I could show you that we provide evidence for evolutionary patterns that parallel those observed in animals and plants, also in microorganisms, and that this community conservatism varies with the ecological specialization and across different taxonomic ranks, offering us insights into the microbial speciation processes and can actually be traced back for billions of years, and that we think that this community conservatism could be a useful signal to potentially refine OTU clustering in the future. With this, I want to thank all the von Mering Lab, the funding, and especially all of you for the attention, and for the MVIF for the possibility to present here. If you would like to, this is our article that is now out in Nature, Ecology and Evolution, if you want to read up on some details. And also, this is the website that you can access to browse around and find or trace your own microbes of interest on our planet. Right. With this, thank you once again.